had a break over the summer of a couple of months. We're just getting used to the technology again today, having forgotten how to use it. But thank you everybody for joining us. My name is Valerie O'Donnell. I'm based in Cardiff in the UK. The title of today's talk is NEO-PUFAs, Isoprostanoids, Promising Oxygenated Metabolites of Omega-3 and Omega-6 PUFAs. Webinar is going to be recorded and it'll be uploaded onto our YouTube channel. It'll be about, as usual, it'll be about 45 minutes. If you would like to ask a question, please put it in the chat or in the Q&A. And at the end, what I'll do is I will call out your name and you can unmute yourself and ask your question and have a chat with Thierry yourself. That's the best way to have a good interaction on Zoom. So, sorry, hang on. Sorry about that. So I'm now going to introduce our speaker. So I'm delighted to introduce Thierry Durand. He studied chemistry at the University of Paris and then moved to Montpellier and received his PhD degree at the University of Montpellier in 1990. After postdoctoral training at FIT in Melbourne with Joshua Rocac, he became Chargé de Recherche, I hope I'm pronouncing that right, CNRS at the University of Montpellier in 1991. He finished his habilitation in 96 and became Director of Research CNRS in 2002. Thierry is a group leader at IBMM Montpellier. His research interests include everything relating to the chemistry of oxygenated fatty acids and PUFAs, I would say. He's provided a list here which is extensive and includes total synthesis of oxygenated cyclic, non-cyclic metabolites of PUFAs, mainly leukotrienes, phyto, neuroprostanes, furans, as well as dihydroxylated PUFAs, branch-chain fatty acids, and many more. I think it's really important to stress how really important to our field, hardcore synthetic chemistry, organic chemistry, and structural chemistry is, how important it is to those of us kind of in the biochemistry and the biology side. It's really important that we work with people like Thierry. I've been fortunate enough to work with people such as Bob Murphy and Alan Brash, and I count Thierry in that ilk of really rigorous hardcore chemists who understand the structures of these lipids really well. And as we move more and more into lipidomics, where we're measuring huge amounts of numbers of different lipids, keeping a really good eye on the rigor of the chemistry that we do is essential. And I think Thierry is a very good example of someone who knows this area inside out. So those of you who might be looking for collaborations in that area, do go knocking on his door, because he's got a brilliant breadth of knowledge across this field in terms of the chemistry of these lipids and the analysis of these lipids as well. And I do want to highlight that back in, I think, 2014, along with a number of colleagues, including Michel Lagarde, he published a seminal paper in Biochemy about, the structural differences between PD1 and PDX, which has really become a kind of a classic in that it highlighted how, easy it is to make a pitfalls in terms of looking at the stereochemistry of really similar lipids, and being able to discriminate these in biological systems is so important, because even though tiny structural changes can look very small, actually they're really important when it comes to signaling and when it comes to biology. So we may hear more about this today, or we may hear about similar stories in relating to other related lipids. I'm very much looking forward to his talk. Thierry is developing collaborations all over the world in different fields linked to oxidative stress, biomarkers, and the biomolecules, which I mentioned earlier. He's published more than 350 scientific articles, reviews in peer reviews, and received the Chevron Medal in 2018 from the SFEL. So many congratulations, and I look forward very much to hearing your talk.
Thank you so much, Valerie, and for your kind invitation first, and also for your kind introduction. And just to mention the importance of the structures of the lipids, but not only lipids. And also I want to thank Caroline for taking care of these seminars, and also Cayman for sponsoring this webinar on lipid maps. So thank you so much. And of course, I will talk today about this NEO-PUFA isoprostanoid. So what we are doing, and of course I am not alone, and it's all my wonderful colleagues. We are co-workers, so we are here, almost chemists and organic chemists, but since a few years, we have also some analytical chemists, because we are doing also lipidomic in this field. And we just moved to a new building we call Institut Ballard. So it's always Institut des Biomolécules Max Bousseron, and he's here, and it's a big building, 28,000 square meters. But we lost a lot of time with that, but it's science, and let's see, we will see. So just what we are doing, and Valerie told you some of them. So we are chemists, and we are doing synthesis of a lipoconjugate. So we are doing some synthesis of lipophenols toward retina pathology. We are doing also synthesis of natural lipophenol, which just discovered recently by my colleague Jetty Lee in Hong Kong, and extra virgin olive oil. And I think we will learn more in a few years on this. We're doing synthesis of lipopeptide with some colleague in Toulouse and discovering microbiotes and targeting pain, and also some lipoxadiazolone with some colleague in Marseille targeting Mycobacterium and tuberculosis. So as you know, we are doing some total synthesis of oxygenated metabolites and called oxylipins, you know well. And we are doing also some dehydroxy-PUFA protectins and others dehydroxy derivative from different PUFA. And more recently, we're doing some synthesis of branch fatty acid hydroxy fatty acid targeting diabetes, but I'm sure other pathology. And as I mentioned also by Valerie, we're doing analytical chemistry, and we do some targeted lipidomic on our isoprostanoid and NEO-PUFA. And recently, Jean-Marie Galano and Claire Vigor also started to work on the molecular networking that all of you are using, and with, I guess, a lot of research in this field. So we collaborate all around the world with people who are interested by our compound, and we publish around 30, 20, or 25, 27, it depends on the Europe, papers per year. And I want to thank all the funders that also support our research. So let's go to the oxidative stress, and you know all of this. ROS can oxidize DNA, protein, lipids, but today I will talk about non-enzymatic oxidation of polyunsaturated fatty acid. So just starting from the beginning of this story, and it's thanks to the Vanderbilt University and Jason Morrow, Jack Roberts, and all these colleagues, just published a PNAS in 1990, end of 1990, just showing that and reporting that there is a high concentration of PGF-like compound in these selenium deficient rats, but in the presence of DQuat or in the presence of carbon tetrachloride and in absence or presence in endomethacin, which just block COX pathway. And the term is compound isoprostanoid. Interestingly, Kadiska published a paper on our FBM in 2005 that's just reported that this F2 isoprostan has to be one of the gold marker of lipid peroxidation. I would say today it's a good markers, but not only one markers. And interestingly, the European Food Safety Authority published in one journal in 2011, so 20 years after the discovery of this molecule from Jason and Jack, and just reporting that it's important now to quantify F2 isoprostan on cardiovascular disease. So what are these isoprostanoid NEO-PUFA? NEO for non-enzymatic oxygenated metabolite of polyunsaturated fatty acid. So arachidonic acid, know, and you know, I guess all of you, as a 15F2T isoprostan. So arachidonic acid led to F2 isoprostan. Linolenic acid, which is present in the membrane of phospholipid in plants, but not only, and I will show you later, produces F1 phytoprostan, and DHA, which is, as you know, in the brain, just produce some F4 neuroprostan. So once again, it's thanks to the Vanderbilt University and colleagues in the Vanderbilt, because they published the pathway of this F2 isoprostan starting from oxidation in phospholipid, okay? And arachidonic acid produced 4 series of F2 isoprostan, the 5, 8, 15, and 12. But later, Yin and collaborator published in GBC that the 15 series and the 5 series has to be one of the best biomarkers and from arachidonic acid. So of course, PUFA and DHA, it's also oxidized and produces neuroprostan and 8 series of neuroprostan. But the same group published in GBC in 2005, that the 4 series and the 20 series has to be the measure of neuroprostan in human. But as you will see, the 10 series of F4 neuroprostan might be also an interesting biomarker in neurological disease. Phytofuran is coming from oxidation of alpha-linolenic acid and just produce only, I would say, the 2 series of phytoprostan, the 9 series and the 16 series. And of course, as prostaglandin, we can find also some different family and rings of the cyclopentan, so it's a well-known. So just to remind you that arachidonic acid produces F2 isoprostan. DHA produces F4 neuroprostan. One interesting PUFA, which is not so a study, is adrenic acid. And thanks again to the Vanderbilt and Van Rollins and collaborators, because they just published that it produced some DHOMO F2 isoprostan. And I will just show you something interesting with these molecules, because it's on the white matter of the brain, contrary to the DHA, which is on the gray matter of the brain. And finally, and I would say not in the, only in the plants, phytoprostan coming from alpha-linolenic acid. So I told you we are chemists, so I will not bother you too much with the chemistry and the total synthesis, but I want just to mention our last strategy, which was developed at that time by Camille Auger, and now she's an associate professor in my team in Montpellier, and Jean-Marie Galano, because he was one of the supervisor of Camille PhD. And this, as you can see, we are able to synthesize a number of isoprostanoids and so on, but not in one step, not in 2 steps, but in 15 or 20 or 25 steps. But hopefully we were able to synthesize some key intermediate, basically key intermediate. And as you can see here, in few steps in a 10-gram scale, and this carbonyl group could be reduced selectively to give and to enter some protective group on the this cyclopentanic compound, and just led to different E and D isoprostanoid. And then when it will reduce completely the carbonyl and the epoxide, we just get some 2-hydroxyl group on the cyclopentan ring, and we were able to synthesize, of course, this DHOMO isoprostan and DHOMO and 7 series of DHOMO isoprostan. But as you can see here in this slide, with this key intermediate, we were able to synthesize a number of isoprostanoids coming from EPA, DHA, DPA, RNA, but also I told you ADA, and more recently, but not yet published, it's from GLA and also SDA. And of course, we pursue to work on this, because more and more we will have standards and reference. We will learn more on these molecules. So we are also engaged in, on the synthesis of this furan and phytofuran, DHOMO isofuran, and neurofuran, because it was also discovered by the Vanderbilt group, and but hopefully with the PhD thesis of Aurélien and also Claire Clermont-Douce, they were able to synthesize this phytofuran and DHOMO and neurofuran, starting from these 2 different strategies and giving the possibility to reach this molecule and to get the standards, and to also look for biological study, because I will show you also a different slide. So the first slide, next slide will be on the biomarkers. I believe it's, and I will try to convince you that it's very interesting to use these biomarkers, not only in human, but in animals and other matrix, vegetables, and more on the algae field. So The first one papers I want to mention is the work from Chiara Carbonondo, and it was published in GBC and also in GR, because Jason and Jack just mentioned that there is some main urinary metabolite of the 15F2T isoprostane. And Valerie mentioned the structure. It was very important because it's some beta oxidation of this compound. And also Chiara shows that and reports that the 2,3-dinor 15F2T isoprostane has to be another metabolite, urinary metabolite of the 15F2T isoprostane. And interestingly, if you look for the 5 series of isoprostane, which has a hydroxyallelic on the upper chain, in this case, like leukotriene and B4, is not beta oxidized. So it's very stable compound in urine. That's why sometimes people quantify the 5 series of F2 isoprostane and found some higher concentration of this compared to the 15F2T isoprostane. So as you know well, better than I, okay, there is 2 main techniques and to quantify this molecule NEO-PUFA, so the GCMS and the LCMS-MS, and more developed the LCMS-MS because it avoids some derivatization and also getting sometimes more interesting data and resolution and separation. So with my colleague Justine Bertrand-Michel in Toulouse, and we were able to quantify and develop a method to quantify around 50 isoprostanoid NEO-PUFA, and in one run, and also sometimes to separate the diastereoisomer. So depending of the configuration of this 8 series of one isoprostane coming from EPA oxidation. So of course we were not alone, and a lot of colleagues and co-workers and collaborators published some very nice method and on this isoprostane field. So Niels Scheb, Maximo Vento and all his colleague and Julia and Consuelo published some interesting on the newborns and also on Alzheimer's. My friend and colleagues, Angel Gil Izquierdo from Murcia in Spain, also published one of the first papers on the quantification of this F1 phytoprostane on extra virgin olive oil. And actually we developed different, and I will show you later. And this one is also recently published and, from Consuela and Maximo group on Alzheimer's disease. And this one is just one papers from JT Lee from Hong Kong. We also developed a very nice methodology. So I want to mention some of them. So this one was interesting because it was one of the first quantification of phytoprostane in human, because it's some work published by Anne Berdin and Trevor Maury in Perth in Australia. So Anne just do some supplementation of flaxseed oil and olive oil and look for different markers. And interestingly, at that time, they just have a high concentration on the phytoprostane on the flaxseed supplemented peoples, contrary to the olive oil. So interestingly also, this formation of this F1 phytoprostane is correlated with the phospholipid inside this patient. So the same group, Trevor and Anne, also published some papers on the interest of F4 neuroprostane on neurological disease, SAH and TBA patient, and also on pregnancy and preeclampsia. As you can see, we have an increase of this F4 neuroprostane compared to normal pregnancy and also compared to the control. This study, and I will just develop a little bit more, because it was done by my colleague Claudio De Felice and Cinzia Signorini in Siena in Italy. And Claudio is an MD and specialist of the Rett syndrome. And they published a nice paper in FRBM in 2009 that there is a link of the oxidative stress and this pathology, because as pathology, it's a rare disease, and it's just affected almost females because it's a mutation on the X chromosome. And what they are looking for, they look for a prognostic biomarkers. And by using our F2-DMO isoprostane coming from adrenic acid oxidation on the white matter, as you can see here, there is a high increase of this F2-DMO isoprostane on the first stage of the pathology, which is not completely developed. And interestingly, not shown in this transparency, but it's on the publication, there is an increase of the F4 neuroprostane depending on the severity of the pathology. So looking for one or another biomarkers, you are able to have some prognostic markers and diagnostic markers. And interestingly, they did some study on supplementation of omega-3 on these small babies and young ladies. And as you can see, after 6 months of supplementation of omega-3, it's a mix of EPA, DHA, we have a completely reduction of the F2-DMO isoprostane. We come back to the basal concentration. And what more interesting, it's some clinical interest also, because this small baby just recovered ambulation because they are not able to walk. And after 6 months, they started again to work with this, and also recovery of purposeful hand movement. They had less hand stereotypies, and the recovery, social engagement, and they try. And after 6 months, they started to talk with their mother and father. That was interesting for them. And of course, there is a decrease of the clinical severity score. This study was done by my colleague Cécile Gladin and John Newman in US, and Cécile is in Clermont-Ferrand in Toulouse, in Clermont-Ferrand, pardon. So they do some supplementation of DHA on this atherosclerotic mice, LDLR minus. And as you can see here, depending on the concentration of the DHA, we have an increase of the F4 neuroprostane. And interestingly, at the time, it was nicely correlated with the formation and the reduction of the atherosclerotic plaque. So at that time, we thought that it could be a relevant therapeutic candidate for atherosclerosis, but nobody took care of this publication, which was published in PLOS ONE. But hopefully we pursue to do this, and I will show you later that it's really a very good bioactive derivatives. This study is also a study from J.T. Lee in Hong Kong. So they look for some rat brain, and as you can see, there is a significantly increased DMO isofuran in this rat brain. Interestingly, if we look at the concentration of the ADA, it's a reduced concentration of ADA, which is perhaps oxidized, but sometimes is not so simple. And there is not always a link between the parent PUFA and the oxidized metabolite of this PUFA. So these two studies, one is from, again, André Gil Izquierdo and Sonia Medina. So it was on an epilepsy patient, and there is an increase of this F2-DMO isoprostane. And this one also published recently in FRBM and by Claudio and Cynthia and his collaborator, Alessio. So they look for different neurological disease, autism, Down syndrome, Rett syndrome, and multiple sclerosis. And as you can see here, we have some specific biomarkers depending on the pathology. And here, as you can see, and I mentioned earlier, the 10 series of 4 neuroprostane was really one good biomarkers in this pathology. So a few study also just on a human milk, and also some study from Maxime Ovento group. So Julia and also Consuelo look for some salivary salivary. So it's interesting if you look some biomarkers. And this one also just published recently on a preclinical AD patient. So as I told you, it's not only on human and animals, but Jetty just also published nice papers on different nuts. So walnut, pine, chia, and flax. And as you can see here, there is a high concentration of F16 phytoprostan and also this 9 phytofuran, depending in the walnut and the chia seed. So this different studies also on vegetables. So interestingly, Handrel, what he did some water stress on olive trees, and he looked on the phytoprostan on olive oil. And as you can see here, there is an increase of phytoprostan in this olive oil, depending on the water stress on the olive trees. This one was done by our colleague Melissa Ioni, and she came in Montpellier, and we did some quantification on melon leaves. And as you can see also, there is an increase of this phytoprostan and phytofuran depending this time thermal stress. Heater by heater, you increase the formation of this oxidized PUFA. And finally, it's also on microalgae, and some colleague in Roscoff in Brittany, and we reported in Food Chem in 2018. And as you can see, with a Cooper stress, we increase the formation of the phytoprostan, phytofuran, and of course, depending on the microalgae, also some isoprostan increase. So I will not, of course, show, but everything is published, and we had just quantified on different grains, seed and nuts, and in almond, in also in mustard wine, in rice, different papers, and also some yes, hazelnut. This one I mentioned already, but this one on cocoa and coffee and co-products. This one on this time on microalgae, and this one recently with Gerard Vandenberg and oxidized fish oil. And we just published one paper 2 days ago with my colleague at France, Frederick Spinner, on pumpkin, and you will just discover this paper on food chemistry. So now I will just talk a few slides on to convince you that this also biomolecules, this NEO-PUFA, is very interesting bioactive lipids. And the first I want to mention is colleague Martin Müller in Germany. So what he did on Arabidopsis. So he did some Cooper stress on this, Arabidopsis, and he looked for if there is a protection on the cell deaths with our phytoprostan. And as you can see here, there is a neuroprotection, I would say, some protection of the phytoprostan in this Arabidopsis. These papers are published by Ralph Bendorf and Edvald Schwelden. So it was not with our isoprostan, it was just commercially available. But what they published in circulation research, a nice paper, that the 15F2T isoprostan possess an anti-angiogenic activity in these human endothelial cells. But interestingly, when we look also the 5 series of the F2 isoprostan coming from arachidonic, it's also possess, of course, less activity on anti-angiogenic activity. But it was the first time that we have some activity on the 5 series of this F2 isoprostan. So this one was done by my colleague Catherine Opéré in Nebraska. So she just looked for biological activity in a retina disease, in bovine retina disease. And as you can see here, there is an effect of this 5-AP, 5F3 isoprostan coming from oxidation of EPA on this potassium evoke release, this neuromediator, which is aspartate release. This study was done by my colleague Luisa Minghetti in Roma, so in Italy. So Luisa looked for some work on the human neuroblast cells, this HSSY5Y. And as you can see here, the phytoprostan are not toxic for these cells. But interestingly, when she did an oxidation with hydrogen peroxide, we have a protection of the cells in the presence of this phytoprostan. And interestingly, she looked for the mechanism and she confirmed by doing some experiment with these nuclear receptors and the 16B1 phytoprostan and those oligodendrocyte differentiation linked to PPAR gamma activation. This study was done again by Cécile in Clermont. So she looked for some anti-inflammatory activity in human cells. And of course, after just introduction of LPS to increase the inflammation. And as you can see here, the 14A4 neuroprostan, which was not done, the synthesis was not done by us, but by my colleagues and friends, Giuseppe Zanoni in Pavia in Italy, and some few know him well. And of course, the 14A4 neuroprostan is more active than the 4F4 neuroprostan, because of the cyclopentanone, which is linked to the NF-kappa B and also linked to the PPAR receptor. But interestingly, these four series of F4 neuroprostan possess also an anti-inflammatory activity, which non-present cyclopentanone, okay? So the next study was really also an interesting study, which has been done by my colleague Jean-Yves Le Guenec and Jérôme Roy during his PhD. Jérôme now is a researcher in RAE, in Saint-Pé, and is working in Truth and with our omega-3 and also with our NEO-PUFA. What Jean-Yves published in 2003 in British Journal Pharmacology, he published that the DHA possess an antiarrhythmic activity, and more interestingly, oxidized DHA had a better activity on antiarrhythmic. And what he mentioned in his conclusion is he mentioned that he wrote that we need to look on oxidized metabolite of DHA, and he mentioned neuroprostan. Hopefully, we met together in a yearly meeting, and I remember, if I remember well, in Rennes, in Brittany. And at that time, he told me that he moved to Montpellier, and I did a presentation, and he did also a presentation. And we said, okay, it will be interesting if you want to test our neuroprostan. Perhaps you will write, and we will just show that this neuroprostan could be active and more active than the DHA. So Jérôme did during his PhD and work on this cardiomyocyte, so on in vitro cells. And as you can see here, there is a nice antiarrhythmic activity of our F4 neuroprostan, more active than the DHA, that's like Jean-Yves thought at that time. And of course, dose-dependent activity, which is interesting. And also, which was validated in vivo model with this PMI mice, because they had some post-infarctus ligated mice. And as you can see here with DHA, but it's the same with our F4 neuroprostan. It was published in FRBM. And what is interesting, it's because this activity is mediated by renin receptors, which is receptors which is present in the cardiovascular disease, but also in a muscular disease. And as you can see here, there is a stabilization of the complex which control this calcium channel, and our 4-neuroprostan just reduce and just possess this antiarrhythmic activity. What is interesting at the time, it's we published, and it was not beta blocker dependent, so because it was not linked to the beta blocker receptors. But at that time, the pharmaceutical company doesn't want to develop this molecule, but that's a problem with sometimes with pharmaceutical company. But hopefully for us, there is one colleague in the labs of Jean-Yves Le Guenec, which is Alain Lacampagne. So Alain Lacampagne was a specialist of this VIEDD. So VIEDD, it's a ventilator-induced diaphragm dysfunction. And when you are a surgery, you pass after a surgery to the anesthesia department to because they maintain your life by doing some mechanical ventilation. And this mechanical ventilation just destroy around 30% of the diaphragm. And after that, people had some problem. Of course, they are alive, but they had some problem. And interestingly, they tested our 4-neuroprostan, an in vitro study and also an in vivo study. And as you can see here, and more, to me, is more fun here because it's restored almost completely this destruction of this diaphragm. And recently, we just confirmed this proof of concept and added pig in Nîmes, a city close to Montpellier with the link to the hospital. And they confirmed this activity of the neuroprostan and better activity on the neuroprostan on this adult pig. So actually, we have some patent and some link to a pharmaceutical company, and I hope perhaps one time we will be able to show you that we are developing this molecule well to drink. So the last study I want to show you today is this study, which is done by my colleague again, Jetty Lee in Hong Kong. What did Jetty did? She looked for biological activity of 4-neuroprostan on the human neuroblast cells, the same cells that Louisa just worked on before and published with in FRBM. But what she did, she looked and after oxidation of these neuroblast cells. And the first thing is interesting, like phytoprostan, the 4-neuroprostan is not toxic for the cells. And interestingly, it protects the cells, not with a big activity, but it's protect from the cells. But interestingly, and more interestingly, what she did an over-oxidation. And after getting these over-oxidized molecules, she got some toxicity in the cells. But hopefully, when you look for oxidation of DHA, you know that it's produced for HHE, and it's known that HHE is toxic for the cells. But the 4 series, if you over-oxidize this, it could be also produced this 4-HHE and started to be also toxic for the cells. But at the beginning, without over-oxidation, with normal oxidation, we produce the 4-neuroprostan and its neuroprotection. So I hope I convince you that this molecule could be interesting biomarkers, and not only biomarkers, but bioactive molecules. So I want to thank first all my collaborators. I mentioned their wonderful job. So JT, Maximo, Andrel, Trevor, Anne, Emily, because at that time she were a postdoc in Trevor and Anne Bardin in labs, but now Emily, she's working in Adelaide Hospital, so always in Australia, and working also on lipid oxidation. Melissa and Monica from Argentina, Chiara, Luisa, Claudio, Edvar, Martin, Nils, Catherine, Gerard, and also Fritz, because I mentioned this nice publication, which will be soon. She's on the web, actually, on Food Chem. And also my French colleagues, so Cécile, Alain, Jean-Yves, Jérôme, Marie, Philippe, Catherine, Thierry from Roscoff, Justine at Toulouse, and Maria and Marina from Montpellier, working on cocoa and also coffee. And of course, I want to, again, to thank you so much for this kind invitation and kind attention. And I will be happy to answer your question if there is some. Thank you so much.
Thank you, Thierry. That was great. And the complexity of the synthetic chemistry always amazes me. It's really heroic work. So very well done. So we have a number of questions. First of all, from Jung Soo. I'm giving you permission to speak there. You should be able to unmute yourself and ask your question.
Oh, hi. A very interesting talk. I was just wondering, is there a gold standard method currently established for LCMS detection of F2 isoprostanes and F4 neuroprostanes? And are there any population-based clinical reference ranges available for those compounds?
Yes, there is not only one method, I would say, because it depends on the apparatus you have. So they first depend also about the tissue and
for fluids, for biological fluids like plasma.
Yeah, biological fluids and asthma. We have some data which has not been published yet, but on different plasma, human plasma. And it's possible to quantify and to find something interesting with this molecule. Yes, of course.
Yeah, so it's not published. It would be awesome if you would be willing to share that somehow.
Sure, we share, as I already mentioned, we share with people who are interested in this molecule. Of course, this molecule are not commercially available, but we can share without any problems and depending of what we have in the freezer, because but this molecule is very, very stable and in the freezer and without any problems. Okay.
Great, thank you very much.
You're welcome.
Thank you. Ginger Milne, you should be able to unmute yourself now.
Thanks, Valerie. Hi, Thierry. That was a great talk. Thank you so much. I have a question for you about omega-3 supplementation in the Rett syndrome study.
Yep.
So in the atherosclerosis study and the anti-arrhythmia biological activities, you see that the biological activity is with the oxidized DHA. Have there been similar studies on this in the Rett syndrome patients? Is it the oxidized omega-3 that's bioactive?
At that time, when Claudio did the supplementation, it just supplemented with fish oil, okay? But at that time, we didn't look on fish oil. But as I mentioned by, which was also published by Gerard Vandenberg, fish oil, if you just quantify, it would contain some F4 neuroprostan. We did something we have not published, but we look for some pills and also that people take for Alzheimer's or so on. That's DHA pills. And these pills contain a large amount of F4 neuroprostan. We published a paper, but it was in chemistry with some, when we did some other series of F4 neuroprostan. And it was also interesting because we quantified this F4 neuroprostan on some omega-3 pills and also some pills from a colleague from Noor industry. Okay, so yes, sure, I guess.
Yeah. Okay, that's interesting. Have you done any biological activities or know of any with the 10 series neuroprostanes? Because I was seeing, we've started to use your great compounds as standards as well, and we do see the 10 series as great biomarkers. So I was just wondering if you studied the 10 series versus the 4 series neuroprostane.
Yeah, it's depend, I will say, of the neurological study. We have also some data not yet published and on isomer patient, different than the one from Maximo group and others. And we had some data interesting with the 14 F4 neuroprostan. We have not yet published, okay? But
okay.
One, as you can see, sometime depending on what you are looking, the interest of doing, I would say, targeted lipidomics is we know where the compound are with the standards, okay? And after that, it depending of the pathology, we could have some prognostic and diagnostic markers depending of the pathology and depending of the series of F4 neuroprostan. Yes, Ginger.
Okay. Thank you again for a great seminar. I think you and I would probably have lots to talk about offline.
Sure. Well,
yeah, thank you, Ginger. Okay, next question from Jose Ordaz Ortiz. You should be able to speak now, Jose. You can unmute.
Hello. Yes. Thank you very much. Yeah, I was wondering then, from the plant-based, we can get that phytoprotein in our diet. Is that correct? And we can, by eating some nuts, for example, we can get a source of F4 phytoprotein, or is that correct? Or no?
Jose, when you hit nuts and so on, you will produce some phytoprotein, okay? And depending of what you have on the nuts or the seed, if you have some amount of DHA, it will be also some production, naturally production of this F4 neuroprostan inside, okay, on the diet, okay? Yes, of course.
Okay, thank you very much. Very interesting talk.
You're welcome.
Thank you. And a question from Christina Ramirez Ferreira.
Hi, thank you for the opportunity to talk with Dr. Durand. So many years ago, we have imaged some ovarian, mammalian ovary sections using the sorption electrospray ionization, and we detected a huge amount of adrenic acid. So at that time, we published this last year, but at that time I was wondering if not only the arachidonic acid, which produces the prostaglandins, but maybe the adrenic acid would be producing some dihomoprostaglandins. I have never found information about that, but I wanted to hear your thoughts if this hypothesis should be considered or pursued.
Thank you, Christina. And I just mentioned this dihomoisoprostan coming from oxidation of adrenic acid. So adrenic acid is oxidized also. I can tell you the percentage of oxidation product, okay? But if you want to detect, and if you look on this oxidized metabolite, I am sure that you will find dihomoisoprostan and dihomoisofuran as well, okay, in this patient. I'm sure but you have to look on. That's a problem, and sometimes people doesn't want to look on. But if you want to look, you can look on this, and you will quantify them.
Thank you.
Okay, so Thierry, I've got a couple of questions myself. And one is that I work more on enzymatically generated oxylipins, of which I guess they're, like a subset of this huge group of lipids that you're looking at. And I'm interested in the different systems that you look at. I guess it varies a lot. What are the relative proportions of these lipids versus enzymatically generated forms that are formed at the same time? In other words, we know that the enzymes that make oxylipins can be a little bit promiscuous, and radicals can leak off the active site, and some of these lipids can get formed through those kinds of mechanisms. And so, it's kind of about the biological origin of them in inflammatory conditions. And, do you measure the enzymatic products like the PGE2, D2, and all the others at the same time, and which tend to predominate? And does that somehow tell you where they might be coming from and how they might be getting made in some of these diseases particularly?
Yeah, Valérie, it's a very, very interesting question. And we had some discussion with colleagues and different congress and so on, of course, of the PGE2 and so on, okay, which is a well-known biomarkers of inflammation. But the problem, and depending of what you're doing on lipid oxidation and also on quantification, sometimes you are not able to separate the 15F2E2 isoprostan or epimer of the 15AP and 15F2E2, sorry, E2T isoprostan and PGE2. So it's complicated because sometimes you have a high concentration of PGE2, and perhaps in this time you have also some isoprostan inside this peak, okay? There is some also some interesting, because some colleagues and Ginger and others, and they just do some ratio on PGF2 alpha and isoprostan depending of the concentration, okay? And sometimes it's depend, and Jean-Marie just describes that in a different review, and it will be a nice review which we publish soon with C-Mondial, and you will see also some interesting things. But yes, it will be actually we just quantify isoprostan, and sometimes we look also for PGE2 depending in the REM, okay? And sometimes we have a high concentration of the 5 series and not PGE2. So it's depend of the pathology, it's depend of the tissue, it's depend of a lot of parameters. But just to keep in the mind that sometime in a peak, you can have different, more than one compound. And that's a problem, I would say, with the PGE2.
Okay, yeah. That's very interesting. I mean, of course, the methods, the reverse phase methods we use have their limitations, and it's really important to be conscious of that and know with a new system, you know, exactly how to look for the structures that you're seeing and what they might be. So we have a question from Pallavi Devchand. I'm just going to allow you to speak, so you can unmute yourself there, Pallavi.
Hi, I was wondering whether you expect each biomarker to be reflective of a single bioactive compound, or whether you're really looking at a biomarker that's reflective of a combination of compounds.
Thank you. It's interesting. At the beginning, we look for biomarkers. So, and a number of colleagues also working in the field of this NEO-PUFA look for biomarkers, okay? And at that time, they didn't think that perhaps it would be some bioactive compound. But if you just quantify some SPM, so enzymatic metabolite of arachidonic and others, okay? You know that there is a really bioactive molecule. So I guess this isoprostan, we didn't test all because it's not possible at present time. But I guess if people want to work with, and we can learn more and more on these molecules. And after that, it will be also to confirm this bioactivity, because you see, Cecile just do something on atherosclerosis, but nobody took care and nobody worked on atherosclerosis. But I'm sure that it would be interesting to test on atherosclerosis, okay? But we have not yet, and we didn't find people we are interested in. But it was very good bioactive compound on the cardiovascular disease, okay?
So I guess what I'm asking is, differentiating the biology from the therapeutic aspect is whether the targets in terms of molecular targets.
Mm-hmm. Yeah, but of course, we are able to quantify, and after that to confirm that it's biological active compound. Yes, of course, it will be very interesting.
That's a great presentation. Thank you.
I mean, in relation to the biological activity, do you think PPAR gamma is a big part of this, just measuring the tone of oxidation in general and responding in a kind of global way?
Yeah, we actually, if I compare to prostaglandin, we have not found some specific biomarkers of isoprostan, okay? I mentioned these renodine receptors, but actually it's not a direct link to the renodine receptors. Perhaps it's linked to some FP or TP receptors. We have some data, I know that Marie and Jean-Yves is working on, but let's see what we will find. But we have not yet some specific markers. I'm not sure if there is, but perhaps not for the 15F2T, but perhaps for the 4 series of neuroprostan. Because this series compared to the enzymatic metabolization is not present on the enzymatic pathway, okay? So that's why it's more interesting compared to the COX. The COX doesn't react with DHA, okay? But the RAS react with DHA to oxidize this molecule.
Yeah, interesting. So I guess one last question before we finish, because we're near the end. I wanted to ask you about dinors and tetranors, which you mentioned at the beginning as metabolites of these things. Do you think they are, biologically inactive, or considering we see quite a lot of those and some in fairly high concentrations for some lipids, could there be more to that story than just non-bioactive end products? Or
we have tested some dinor and tetranor and found some biological activity, not would say less active than the 15F2T, but interestingly, I can mention you this story also because you mentioned about PD1 and PDX, okay? We published a nice paper with my colleague Angelo Sala in GMedChem last year or in 2019, I don't remember, but about dinor and tetranor metabolite of PD1. And this dinor and tetranor is also active and of course, less active than PD1, but it's also active. So sometimes depending on the biology, you inject the compound, but perhaps it's the dinor or others metabolites, of course.
And then it really depends on the concentrations of the relative concentrations, which one is active. So, okay, so I think we've run out of questions and we're coming up to the end anyway, so perfect timing. And I want to say thank you very much again, Thierry, and to Cayman for sponsoring this seminar. Thank you for a wonderful webinar, and we look forward to seeing everybody again for the next webinar in I think 1 or 2 months from now, which will be advertised soon, which is going to be Richard Gross, I believe. So thank you everybody again, and see you all soon. Goodbye.
Thank you so much. Bye-bye. Thanks a lot.